Hi everyone, this is lecture 26 and we're going to be talking again about independent assortment. Today we're going to learn a new technique called the forked line method for solving independent assortment genetics problems. Alright, so the material here comes from chapter 11, page 307 to 312. The first question, full pod shape F is dominant to constricted pod shape lowercase f, and yellow pod color capital Y is dominant to green pod color lower y in pea plants. A pure breeding green plant with full pods is crossed with a pure breeding yellow plant with constricted pods. What will be the phenotype of the offspring? Number 2, assuming that all of the genes in the following example assort independently, what proportion of offspring from a cross between two big A little a, big B little b, big C little c, big D little d, big E little e, and big F little f individuals would be big A, big B little b, big C, little d, big E, and big F little f. So from last time we saw a dihybrid cross. A dihybrid cross is when you cross two individuals that differ in two traits, not just one. For example, pea plants that have not only the yellow and green color, but also the smooth and wrinkled texture. The yellow allele is capital Y, and the green allele is lowercase y. The smooth allele is big S, and the wrinkled allele is little s. These are denoted by the colors on the chromosomes here. When you cross two of these true breeding plants together, you end up with hybrid in the F1 generation, which are all yellow and all smooth, as shown down here at the bottom. When you cross two of these hybrids together, you generate the 9:3 to 3:1 ratio. The gametes that are formed are shown here on the bottom. Some gametes will have both the smooth and yellow alleles, some will have the smooth and green allele, some will have the wrinkled and yellow allele, and some will have the wrinkled and green allele. Crossing them together in a Punnett square, again, gives you the 9:3 to 3:1 ratio. As you can see from the picture here, each one of these along the x and y axis is a gamete. They have equal probability of fertilizing each other, so that the zygotes that are formed are shown in these 16 squares. You can express these 9:3 to 3:1 ratios as fractions. So of the 16 total possible offspring, 9 of them are smooth and yellow, 3/16 are smooth and green, 3 out of the 16th are wrinkled and yellow, and 1/16 is wrinkled and green, both recessive. Again, yellow is dominant to green, and smooth is dominant to wrinkled. This kind of Punnett square is a little bit cumbersome. It's challenging to get it absolutely correct. It's easy to make mistakes, as you can see from the number of squares you have to fill out. Also, on a test situation, this kind of Punnett square would take you an inordinate amount of time to finish. So we want a simpler, maybe an easier way to do this. And that method is called the forked line method. The forked line method works like this. You break it down into each individual trait. Remember, these two traits, the color and the texture of the seed, sort independently, so we can treat each one separately. For example, we'll start with the texture, the smooth versus wrinkled. We know from a monohybrid cross of these traits that smooth is dominant to wrinkled, and so will appear in a 3:1 ratio. 3/4 of the offspring would be smooth, and 1/4 would be wrinkled. We denote the smooth genotype as big S with a dash, and the reason for that is that the dash indicates that the smooth could be true breeding, big S, or they could be heterozygous, big S, little s. It doesn't matter for this forked line method. However, for the genotype for the wrinkled seeds is absolutely known. It's little s. That's the only genotype that can produce that phenotype. For the second trait, we do the same thing. Yellow versus green. 3/4 of the offspring in a monohybrid cross would be yellow, and 1/4 would be green. Again, that's the Mendelian 3:1 ratio. 3/4 are big Y with a dash, again because some of them are big Y, and some are big Y, little y. but the green ones are absolutely little y, little y. What we're doing here is we're multiplying fractions together. That's it. It's very simple. If you can multiply fractions, you can do the fork line method. We're assuming that fertilization is completely random so that each smooth seed has an equal probability of matching up with a yellow or a green. That's why this fork line is shown equal, one this direction, one this direction. So you simply, to figure out the ratio that are smooth and yellow, we multiply. And it's kind of like a probability. Whenever you use the word and, when you're talking about probability, you are multiplying. You want to know how many are smooth and yellow. So you multiply 3/4 that are smooth times 3/4 that are yellow for 9/16 that are smooth and yellow. The genotype here is shown big S with a dash and big Y with a dash. We don't really care about the genotypes in this situation. We just want to know what the phenotype ratios are. To figure out how many are smooth and green, we simply multiply 3/4 times 1/4 for 3/16. If we want to know how many are wrinkled and yellow, we multiply 1/4 wrinkled times 3/4 yellow to get 3/16 yellow and wrinkled. And finally, to figure out how many are wrinkled and green, we multiply the 1/4 wrinkled times 1/4 green to get 1/16 wrinkled and green. You'll notice this is not a Punnett square, but we get the same ratios, the 9:3 to 3:1. And this is much more amenable to more complex genetics problems where the Punnett square becomes very cumbersome. All right, we'll practice these in class, so I will see you then. Bye-bye.